Hello everyone. Uh, today's today's topic is nucleic acid, and hello Gaurav. Today's topic is nucleic acid and complex carbohydrates. So this is a continuation of the biomolecule class. So what is nucleic acid? So first topic which we will cover is nucleic acid. I hope my voice is clear. Okay. So nucleic acid, if you look at nucleic acids are basically a type of macromolecules. First thing we need to understand that nucleic acids, they are a type of a macromolecule. What is a macromolecule? A macromolecule is something which has a molecular weight over 1000 daltons. Dalton is basically a unit to measure any kind of biomolecules. Okay, anything which is below 1000 daltons is called micromolecule. Anything above 1000 dalton is called a macromolecule. Examples of macromolecules are nucleic acid, as I said. We also have protein. Okay, and we have polysaccharides. Okay, polysaccharides, just say we have starch, we have glycogen, then we have cellulose. Okay. In my previous lecture, we have discussed about the structures of polysaccharide in details. Okay. So nucleic acid is also a it's also a polymer. Okay, it's also a macromolecule. And when we look at a nucleic acid, a nucleic acid is, we have two kinds of nucleic acid. We have the RNA and we have the DNA, right? So this is called the ribonucleic acid, and this is called the deoxyribonucleic acid. Okay, so there are two kinds of nucleic acids, RNA and DNA. So nucleic acid is made up of polynucleotides. Poly means many. Okay, many nucleotides. So we need to understand what is a nucleotide and how many nucleotides they join and form a polynucleotide, which is basically the nucleic acid. Okay. So let's go to the notes. And first thing, so this is basically a polynucleotide. So if you can see, Gaurav, each of this. So this is a nucleotide. This whole thing is a nucleotide. Why it is a nucleotide? Because
Sir,
a nucleotide is made up of, so when we go to a nucleotide
hello, sir.
is, Gaurav, you want to say something?
Yes, sir.
Yes, go ahead.
Sir, what is the difference between DNA and RNA?
I will come to that. First, we will learn step by step. Okay. First, we'll learn what is a nucleotide, what's a nucleoside, and after that, we will go to automatically. We'll understand what's the DNA and RNA difference. Okay. So we'll go step by step. So first, nucleotide is basically, it is made up of, number one, it has a sugar. Okay. then it has a nitrogenous base, and finally it has a phosphate. Okay, so these three components make it a nucleotide. So now, if we take these two components, sugar and nitrogenous base, any idea what is this called, Gaurav? Sugar and nitrogenous base? It is called a nucleoside. Okay, just if you take the sugar and nitrogenous base, it is called a nucleoside. If you add a phosphate, it becomes nucleotide. A DNA and an RNA is polynucleotide. That means many of these nucleotides, they join by a phosphate and a sugar phosphate backbone. Okay, a sugar phosphate backbone, and it forms a, we'll talk about that. We'll talk about all those things in details. So now, let's go to the structure of this sugar, nitrogenous base, and phosphate. And one thing I think most of us know, that a nitrogenous bases are basically, we have two kinds of nitrogenous bases. We have the purines and we have the pyrimidines. Purines are basically adenine and guanine. Are you familiar with all these things, Gaurav? These concepts? And pyrimidines, we have thymine, we have cytosine, and we have uracil. So there are three kinds of pyrimidines and two kinds of purines which make up the nitrogenous base. Why it is called nitrogenous base? Because these are heterocyclic compounds. Hetero means different kind of elements are there. Okay, heterocyclic. They are mainly made up of nitrogen. Okay, so they have rings. Okay, they have aromatic rings. Heterocyclic. Cyclic means rings, right? And they have nitrogen, hydrogen. Okay, and different kinds of other kind of elements are there. All right, so that's why they call heterocyclic. The principal thing is nitrogen in these nitrogenous bases, and these nitrogenous bases are an integral part of our DNA and RNA. Okay, all right. So now, when we go to so here, now when we look at a nucleotide, this is basically the nitrogenous base. This is a sugar, and this is a phosphate. All three of them together, we call it as a nucleotide. So let me try to draw this. If I take a section here, this is actually the nucleotide. Again, with a different color, this is another nucleotide. You can see this is a nitrogenous base, this is a sugar, this is a phosphate. Okay, so this is another nucleotide. Okay, then we have another nucleotide. Here is a nitrogenous base, this is the sugar, it's a phosphate, right? So you see one nucleotide, two nucleotide, three nucleotide, four nucleotide, and this is a polynucleotide, which is one strand of a DNA. This is a one strand of a DNA. Okay, so now more details we will get into this thing. So this is basically how a DNA looks like, a double helical structure. Okay, right? Each strand is made up of many nucleotides. Each nucleotide is made up of three components: a nitrogenous base, a sugar, and a phosphate. And it is joined by a phosphate bond and a sugar phosphate bond. So what is the sugar phosphate bond? So this is the sugar, this is the sugar, this is the phosphate. Right? This is called the sugar phosphate backbone. Sugar phosphate bond, sugar phosphate bond. You see that? Sugar and phosphate, sugar and phosphate. This is called the backbone of a polynucleotide. I hope Gaurav, you're understanding these concepts. So now we go into more details about the structures. Here, this is actually a very clear picture of a nucleotide. And as you can see, this is the nitrogenous base, which is called a heterocyclic compound because it has two aromatic rings. Okay, so this is an example of purine. This is example of purine. This is called adenine. Okay, you see this is a heterocyclic compound. This is also called it's called a nitrogenous base. Why we call this as a base? What is the base thing here? A base is something that acquires hydrogen. Understanding? Any base is something that acquires hydrogen. Anything that is acidic that loses hydrogen. This is the basic concept of chemistry, right? So as you can see, this nitrogen here, it is acquiring these two hydrogen. Understanding all of this concept? That's why it is called a nitrogenous base. Base is always positive, right? Basic is positive, acidic is negative, right? So when we go to actually these higher kind of classes, 11, 12, okay, when we are targeting the NEET, okay, these kind of concepts are very important. Why do we call this a nitrogenous base? First of all, it has lots of nitrogen in this heterocyclic compound, and it is also binding to this hydrogen. That's why it is called a base, okay? Now, next is a sugar. You see, this is actually a pentose sugar. Any student who has attended my carbohydrate class, they will understand what is a sugar. You see, it's a hydrocarbon. That means carbon and hydrogen. Each of these dots, okay? So each of these dots is a carbon. You see that? Each of these, so this is 1, 2, 3, 4, 5. This is the fifth carbon. This is the fourth carbon, third carbon, second carbon, first carbon. Okay, so this is basically a ribose sugar. Ribose sugar. Why ribose? Because there is 5 carbon. It's called a ribose sugar. Okay, and this ribose sugar, here you ask me a question, okay, what is the difference between a DNA and an RNA, right? So you see at the second carbon here, there is a H group. There is a H group. If you look at RNA, at the second carbon, there will be a hydroxyl group instead of a H group. So that's why DNA is called deoxyribose. So let's see a picture of RNA and DNA. That will clear the whole concept. So here is the picture of a sugar that is present in RNA and sugar that is present in the DNA. So here you see this is a sugar that is present in the DNA, and this is the sugar that is present in the RNA. Look at the second carbon. At the second carbon, there is a hydroxyl group. Can you see that in RNA? Okay, so when we draw a sugar of a RNA, this is how we draw. Okay, we have 5 carbons, 1, 2, 3, 4, 5. Okay, and it has this pentagon shape, right? Pentagon shape. There is oxygen here. Okay, and you see there is hydroxyl group, hydroxyl group. If you look at the DNA, there is a hydroxyl group in the first carbon. In the second carbon, instead of a hydroxyl group, there is a hydrogen group. So that's why this is the deoxy. Deoxy, that means one oxygen is absent, right? Deoxyribose. It is a ribose sugar because it's a 5 carbon sugar. It's a 5 carbon sugar. It's called a ribose sugar. Okay, it's a ribose sugar. And this ribose sugar has one oxygen less. So it's called deoxyribose. I hope, Gaurav, you can understand the concepts. Okay. In case of RNA, the second carbon has a hydroxyl group. It has a hydroxyl group. So that's why it is called ribose. Just plain ribose sugar. This is called deoxyribose sugar because one oxygen is gone. Okay. So in DNA, in the nucleotide of DNA, we have these deoxyribose sugar. In the nucleotide of RNA, we have these kind of ribose sugar. All right. Again, here we can see the different kinds of the nitrogenous bases which we see in DNA and RNA. So as you can see, this is adenine, this is guanine. These two things are called the purines. They're called purines because they have two kinds of cyclic rings. See, one ring, two ring, one ring, two ring. There are two heterocyclic rings in the purine. If you look at the pyrimidines, pyrimidine has only one heterocyclic ring. Okay. and this is thiamine. Look at thiamine. Thiamine has an extra methyl group. Okay, this CH3 group, it is called a methyl group. CH3 is a methyl group. When you look at uracil, it doesn't have this methyl group. Okay. In case of DNA, DNA has thiamine, RNA has uracil. RNA, you will never see thiamine. Okay. You will always see uracil. Okay. And we know A adenine pairs with thiamine, guanine pairs with cytosine. This is called the Chargaff's rule. Right? Chargaff's rule. It's called the Chargaff rule, that adenine always pairs with thiamine and guanine pairs with cytosine. In case of RNA, adenine pairs with uracil. That's why thiamine and uracil have more or less the similar structure. Only difference is that in case of thiamine, we have a methyl group. In case of uracil, the methyl group is missing. Other than that, if you look at if you compare the structures, you see they are kind of similar kind of structures, thiamine and uracil. Okay. This is cytosine, and cytosine has this amino group present, okay, with this carbon. Cytosine binds with guanine, right, in case of in a DNA, and in an RNA. And RNA is normally single-stranded, DNA is normally double-stranded. So in case of a DNA, in the two strands, the guanine and the cytosine, they pair, adenine and the thymine, they pair. So going back to the DNA structure, now, Gaurav, you will understand that how, so now we know. So let's see now what kind of
Yes.
concepts we have realized. Yes, Gaurav, go ahead. Any questions? You can go ahead. So now this is
Yes,
basically
sir.
the, yes, Gaurav, go ahead. Tell me.
I understand, sir.
Understand? Okay, good. So now this is the two strands of DNA. Now, Gaurav, it becomes very, very clear. So this is a nitrogenous base. Tell me, what is this nitrogenous base? It has two rings, right? So what is this base now? Now what we have learned from this lecture, this is a purine or pyrimidine. What is this? It has two rings. It is it's a purine, right? It is a adenine, okay, because it has two rings, right? Unlike, look, this one has one ring. This is a cytosine, right? Again, this is one ring. This is a thymine. This is two rings. This is a guanine, right? So we know in purines have two rings, pyrimidines have one ring, right? So this is bonded with the sugar. And now we know, okay, this is a DNA, and DNA has a deoxyribose sugar. Yani ki there is only hydrogen. There is no hydroxyl, right? So this sugar is bonded on one side with the nitrogenous base. This same sugar is bonded with the other side with the phosphate, right? So we have a nitrogenous base, a sugar, and a phosphate. This becomes one nucleotide. Again, the cytosine is bonded with a sugar and a phosphate. So this becomes another nucleotide. Here, this becomes another nucleotide. This becomes another nucleotide. This is how we have one strand of DNA, which is polynucleotide. Very, very important. There is always a sugar and a phosphate backbone. There is always a sugar and a phosphate backbone. Here you see sugar and a phosphate, sugar and a phosphate, sugar and a phosphate. Okay, this is the sugar phosphate backbone. Just like in a human, we have a vertebral column, right? In a DNA strand, this is their vertebral column. This is their backbone. Okay, so always we have a sugar and phosphate backbone, which is actually forming the DNA strand. Okay, and DNA has two strands. You see, there are two strands. They are bonded by hydrogen bonds. Can you see that? Can you see how this hydrogen and this oxygen, they're bonded by a hydrogen bond? Here, this and this, they have a hydrogen bond. This hydrogen bond is keeping the two strands of DNA attached to each other, and it's forming this kind of helical structure. Here, we have this kind of helical double helix, right? This helical structure it is forming. Okay, so to draw this better, this is actually how the helical structure looks like. So parallel helical structure, and in between we have these kind of hydrogen bonds holding the two strands together. Okay, these two strands are anti-parallel. They are anti-parallel. That means this strand is 5' to 3'. Okay, what is this 5 dash 3 dash, Gaurav? This 5 dash is basically, look at the carbon number in the ribose. Very, very important. If you look at the carbon number, this is the first carbon, second carbon, third carbon, fourth carbon, fifth carbon, right? So this is the fifth carbon attached to the phosphate group. All right. And this 3 carbon is attached to the phosphate group. Can you see that? So 5 dash 3 dash. Again, here in this sugar, we have the fifth carbon attached to the phosphate group. Here, the third carbon of the ribose is attached to the phosphate. So it's 5, 5, 3, 5, 3, 5, 3. Understanding? 5, 3. So we call it 5 dash to 3 dash. Right?
Yes, sir.
Makes sense? The strand is 3. I mean, look at the opposite strand. Here it is running opposite. 5, 3. Right? Look at how the sugar and the phosphate they're attached. Here the fifth carbon is attached to the phosphate. Here the third carbon is attached to the phosphate. Again, fifth carbon to the phosphate, third carbon to the phosphate, right? Fifth carbon to the phosphate, third carbon. So this is 5, 3 in this direction, right? You see it's opposite, 5, 3 this is, and this is 5, 3 in this direction. This is called anti-parallel strands. We call this anti-parallel strands of the DNA. Clear? So now we know that DNA is made up of nucleotides. What are nucleotides? Nucleotides are made up of nitrogenous bases. They are made up of sugar. They're made up of phosphate, right? And so this is one actually nucleotide. DNA is a polynucleotide. DNA and RNA are macromolecules. Okay. What are macromolecules? Macromolecules are something which has a molecular weight higher than 1000 daltons, right? Lipid, if you look at, lipid is not a macromolecule. It's a biomolecule. Lipid, okay, I've talked about lipid in other one of my other lectures. And lipid is basically, it's actually technically a micromolecule. Lipid size is less than 1000 daltons. But if you look at protein, if you look at polysaccharides, if you look at nucleic acids, okay, these biomolecules are biomacromolecules. What do we call them? Biomacromolecules.
Molecules.
Why macro? Because their size is more than 1000. But lipid is not a biomacromolecule. It is simply a biomolecule, right? Because lipid has its molecular weight is less than 1000 daltons. Okay. All right.
So what is unit Dalton?
Dalton is a unit, just like all these biomolecules we measure by daltons. Okay. We don't say kgs, we don't say grams or pounds. Understand? Okay, so we using Dalton is basically a unit to measure these kind of molecular compounds. Make sense? We can't use a kg. We can't say 10 kgs, 1 kgs. We use the word Daltons. That's universally accepted kind of unit. Okay.
Sir, how
Or
can we definite a Dalton?
how can we differentiate Dalton?
Definite.
Definite means got of one thing you have to understand that these are very, very small, right? They're present inside the cell. Okay. So inside the cell they are present. They are sub cellular or sub microscopic, right? uh, we can't look at these under normal microscopes, right? So they're sub microscopic. So that's why their unit is given a special unit to actually measure macro biomolecules. Understand? This is a unit to
Okay, sir.
measure biomolecules. You can't use pound and kgs, and they can't go so less, right? These are
Yes, sir.
subcellular. Understand? So here the scientist had to give a very special kind of unit which can measure these very, very small actually biomolecules by using the word Daltons. Okay. So till
Okay,
now
sir.
at this level, I think this is enough. We don't have to go into details of how Dalton and how why it is given. Just know okay, any kind of bio macromolecule has 1000 Dalton above. Any just simply biomolecule or bio micromolecule, their actually weight is less than 1000 Daltons. Okay. All right.
Okay, sir.
So now I think we have finished all about nucleic acids. Now we'll go to complex polysaccharides. So what are complex polysaccharides? In my last lecture, Gaurav, if you have attended that, there I've talked about polysaccharides in general. Just a small brush up of what I've done last class. So basically, when we talk about polysaccharides, so these are the different kinds of polysaccharides. Okay, just refresh. So this is basically glucose. This is the 6 ring of glucose. Okay, you can see carbon 1, carbon 2, carbon 3, 4, 5, and 6, right? This is how we write a glucose. And glucose has lots of isomers, right? Look, if you look at glucose here, you see glucose has hydroxyl group, and the carbon 1, the hydroxyl group is at the bottom of the plane, right? If this is the plane, it is the bottom of the plane, right? This is also bottom of the plane. The third carbon, look, the third carbon, the hydroxyl is above the plane, right? You see that? Again, the fourth carbon is below the plane. This is the plane. Okay. Now, when you look at galactose, galactose has the fourth carbon, which is above the plane. Look at glucose, and if you compare glucose and galactose, right? You see the fourth hydroxyl group is above the plane. Here, the fourth hydroxyl is below the plane. That is the only difference between glucose and galactose. Otherwise, their molecular formula is same. C6H12O6, right? They have 6 carbon, 12 hydrogen, 6 oxygen. So molecular formula is same, but the orientation is different because in galactose, the fourth hydroxyl group is above the plane. Here it is below the plane. Understanding? They are called isomers. All right. Same concept. Fructose and glucose are also isomers. Why are we studying these? These are all carbohydrates, right? And they're all monosaccharides because they are made up of only one unit. It's called monosaccharides. So glucose, fructose, galactose, okay, mannose, they're all monosaccharides. Okay. Look, this is actually fructose, and fructose has a different kind of arrangement. This hexagonal ring, this is called pyranose. What do we call this? Pyranose arrangement. This hexagonal ring, where we have 6 actually, right, points. This ring is called the furanose arrangement, right? Furanose arrangement. Look, 1, 2, 3, 4, 5 here. Although there are 6 carbons, because this carbon 1, 2, 3, 4, 5, 6, but the ring has only 5 carbons. So it's called a furanose arrangement, which we see in case of fructose. Fructose and glucose again are isomers. Same kind of molecular formula, C6H12O6. But look at how the arrangement is different, right? We see how this kind of arrangement or the spatial arrangement is different. Okay. So they are isomers having different spatial arrangements, but same kind of molecular formula. When we look at disaccharides, look, this is sucrose made up of glucose and a fructose. Can you see that? This is the glucose, right? And this is the fructose. They're joined by this kind of bond, 1,4 linkage. So no, this is actually, it's not a 1,4 linkage. This is the 1,2 linkage. This is not a 1,4 linkage. I'll just leave this. So this is called a 1,2 linkage. Why 1,2? This is the first carbon. This is the second carbon. Okay. So this is a 1,2 linkage in case of sucrose. And this is lactose. Lactose is made up of one glucose and galactose. Can you see that? This is the galactose. The hydroxyl group is above the plane, right? And this is the glucose. So this is a 1,4 linkage, 1 carbon, 4th carbon, 1,4 linkage. It is called the lactose. Lactose is a disaccharide. Sucrose is a disaccharide made up of 2 monosaccharides. Clear? And last class also I explained what is a reducing sugar, if you have followed my last lecture. And we have also talked about what is a non-reducing sugar. And I've explained why lactose, which is a disaccharide made up of galactose and glucose, why it is a reducing sugar, because it has one free anomeric carbon. Okay, I'm not going to explain that in this lecture. In case of sucrose, both the anomeric carbon, they are engaged in a bond formation. What is an anomeric carbon? It is the carbon that is bonded to the oxygen and the hydroxyl group. That's called the anomeric carbon. Actually, in a very simple way, it is the carbon number 1. which is bonded to the oxygen and the hydroxyl. That's called the anomeric carbon. So here you see, this is the anomeric carbon. So this is the anomeric carbon. This is the anomeric carbon bonded to the oxygen and the hydroxyl group, right? And in case of fructose, this is the anomeric carbon bonded to the oxygen and the hydroxyl group. Okay, so this, in case of sucrose, we can see this anomeric carbon is exchanged in a bond formation. It is involved in a bond formation. It is not free. That's why it is a non-reducing sugar. And in case of lactose, this anomeric carbon is free to react with another molecule. That's why this is a reducing sugar. Why reducing? It can reduce a cupric to a copper into a cuprous state. Cupric is a 2+ state of copper. It can reduce it to a cuprous state. Okay, all these things I've explained in my last lecture. So now, today we will go to the concept of complex carbohydrates. Complex carbohydrates are carbohydrates with some kind of complex group attached to that. Okay, so let's see what a complex carbohydrate looks like. And remember, Gaurav, all these things are actually included in the NCRT, right? In class 11, 12, in the NEET. Okay, all these kind of questions, they come. That's why I'm teaching it. So this kind of lecture will help you in the NEET examination. Okay. So look, this is a normal glucose, right? This is a normal glucose. But look at this. This carbon is bonded to an amino group. Can you see that? This is amino group. So this is called the glucose amine. Can you see that? Why it is glucose? Because this is a glucose structure. Look at the hydroxyl group. Third carbon hydroxyl group above the plane. Fourth carbon hydroxyl group is below the plane, right? So this is glucose. This is galactose, right? Fourth carbon hydroxyl group above the plane. This is galactose, isomer of glucose. Look at the second carbon bonded to the amino group. It's called galactosamine. Okay, glucosamine, galactosamine. So complex carbohydrates. I hope you're understanding the concept. Complex. So sometimes with this NH group, a COCH3 group can be attached. This is called the acetyl group. Okay, CH3CO is called a acetyl functional group. Understanding? So this is called, so once that is done, it is called a N-acetyl glucosamine. Look, if there is only an amino group, it's called glucosamine. Now you add a acetyl group, CH3CO. You add a acetyl group. It's called N-acetyl glucosamine. Okay, initially it was just glucosamine. Now you add an acetyl group. N-acetyl glucosamine. Why are we studying this? It's a complex carbohydrate. Where do we find this? We find it in the chitin that is present in arthropods. Arthropods is covered by a chitinous layer. Okay, if you see any kind of arthropod, like a beetle, like an insect, like any kind of grasshopper, their body is covered by a tough chitin layer, right? So this is the chitin formula. This is the molecular formula of chitin. It is basically glucose, but there is actually a N-acetyl glucosamine attached to the carbon ring. Understanding? A normal glucose, how will it look like? So when we draw a normal glucose, so that all the concepts are clear, you see a normal glucose will have a 6 carbon, right? And I'm drawing in blue. So this has a hydroxyl, a hydroxyl, a
Sir,
hydroxyl.
why we add in
Yes.
acetyl group?
What, Gaurav, what is the question?
Sir, why we add N in acetyl group?
Why do we add N? Because the N signifies, this is the N, okay? All right. So this is the nitrogen, the N-acetyl glucosamine, okay? Because the acetyl group is attached to the N of the amine group, so N-acetyl glucosamine. Makes sense? So that is basically the
Okay.
composition of arthropods. Even in fungi, if you look at fungal cell wall, okay, it also has chitin in the cell wall. Okay, it doesn't have a normal cellulose. It has a chitinous cell wall. composition. Makes sense? So these are basically types of complex carbohydrates which we see in different kind of applications, right? In arthropods, in fungal cell wall. Okay. So, all right. So what we'll do, Gaurav, is we'll stop here. This is the end of the 40 minutes. Okay. I hope this lecture was helpful because we learned about nucleic acids. We learned about what are nucleotides, right? Polynucleotides. We learned about the pyrimidines, purines, right? We learned about hydrogen bond formation in the DNA, and then we learned about carbohydrate basics and also about the complex carbohydrates. Any questions you have, Gaurav, in today's lecture?
No, sir.
Okay. So I hope these Bio-kul classes are helpful. Okay. Keep attending these classes. And again, I will upload the recorded video and I will also post some polls, right? 5 poll question. I hope you will be able to answer them because you attended the live class, right?
Okay, sir.
Okay. Again, tomorrow I'll come up with actually a new content. I will also do class 9, class 10 contents. Okay. Right. The CBC is approaching. So I will have these kind of crash courses, right? In one class, I will finish one topic. All right.
Thank you, sir.
Okay. Welcome. Bye bye. See you tomorrow.